from the National Institute of Respiratory Diseases from Mexico City. And my work is entitled MiRNAs involved in the signaling pathways associated to the pathogenesis of idiopathic pulmonary fibrosis. I don't know why my presentation can be shared.
Dr. Cadena, in the PowerPoint, it says click habilitar edición.
Okay.
Okay, you're welcome.
It's opening. I'm sorry for the delay. Okay. So okay. Can you see the presentation, right?
Yes.
Okay. Can you see the pointer too?
Yes.
Okay. Well, thank you. I will start by explaining a little bit of idiopathic pulmonary fibrosis, which is a chronic and progressive disease with poor prognosis and few therapeutic options. And it is characterized by epithelial disorder and for collagen deposition. And it's because when there is an epithelial cell injury, there is a formation of a lung scar, which leads to excessive fibroblast activity and also to fibroblast transdifferentiation. And there is a process which is the main process found in this disease, which is epithelial mesenchymal transition. And this process is characterized by the transdifferentiation of fibroblasts into myofibroblasts, which are the cells which synthesize excessive collagen. And there is also a resistance to apoptosis and matrix deposition, like it's seen in the image. So what is a microRNA, which is abbreviated MiRNA? MiRNAs are long coding RNAs, and they are in charge of post-transcriptional regulatory role and the regulation of cellular processes such as apoptosis, proliferation, and cell differentiation. And these MiRNAs are found to be dysregulated in idiopathic pulmonary fibrosis. As I said, the epithelial mesenchymal transition is a very important process in this disease. because it's characterized because epithelial cells gain new invasive properties and increase extracellular matrix proteins. And this generates the lung scars and the disease by itself. So there are a lot of signaling pathways in this disease, like they have been studied in this disease, but the most important ones is the signal pathway that involves the transforming growth factor beta 1. And this factor stimulates a SMAD pathway and a non-SMAD pathway. In the non-SMAD pathway, we can find the W and T and the phosphatidyl inositol kinase signaling pathway. So there is a crosstalk of signaling pathways involved in the epithelial mesenchymal transition process. And the most studied ones is the one that involves TGF, because TGF beta is the most important one. And this signaling pathway can be activated by interleukin 1 and insulin growth factor and some other substance, but the most important ones are these ones. And when they make contact with the receptor of TGF, it can follow a SMAD-mediated signaling pathway or a non-SMAD mediated signaling pathway. And when they follow the SMAD pathway, when the receptor activates, it phosphorylates SMAD2 and SMAD3. And these two forms a complex that travels from the cytoplasm to the nucleus. And they make contact with SMAD4, and they through SNAIL1 and 2 and SEV1 and 2. And together this complex forms, well, they make possible the EMT, the epithelial mesenchymal transitions. Also, they regulate profibrotic genes, and they regulate collagen expression, and also fibroblast differentiation and proliferation. And they are like antifibrotic agents, profibrotic agents, because they regulate these processes. But also we have a contra-regulator, which is SMAD6 and 7. This complex regulates the SMAD2 and 3. So they are considered, this complex is considered anti-fibrotic complex. And also we have a non-SMAD mediated pathway, which is led by MAP kinases. And because this kinase can activate RAS and ERK, and also the TAC1, and they can also mediate the SMAD2 and 3 complex. So they also start this signaling pathway, and they favor the epithelial mesenchymal transition, the profibrotic genes, and the collagen expression. And there are some other non-SMAD mediated signaling pathways, which are phosphatidyl inositol kinase, which activates the AKT and FOXO3. FOXO3, it's a mediator which has the function of regulating P27, which is a kinase that regulates the cellular cycle by inhibiting this cellular cycle. And specifically, it acts through the G1 process of this cellular cycle. And also, phosphatidyl inositol can activate mTOR and mTOR1 and mTOR2, which are very important because they regulate the fibroblast differentiation and the epithelial mesenchymal transition too. And finally we have the WNT pathway, which is activated when it forms a complex with the Frizzle family from 1 to 10. And they, in a steady way, it is inhibited by axin 1. And but when it binds to the Frizzle family, it activates, and the function is to activate beta-catenin and some other signaling proteins, but they have an impact on collagen genes and metalloproteinases. So MiRNAs can act as profibrotic mediators or antifibrotic mediators because they can act in the receptors of these signaling pathways. And we will see some examples in the next templates. For example, we have, this table is illustrating MiRNAs involved in the epithelial mesenchymal transition in a SMAD-dependent way. And these MiRNAs has been studied in both human models in bronchoalveolar lavages, but also from patients with IPF, but also in bleomycin murine models, which is a model in which mouses are injected with bleomycin, which creates a fibrotic lung, and also in some cell lines like A549. So, for example, in the first one, we have the bleomycin, the sample is bleomycin murine model, and the studied MiRNA is the 877 3P. And his function in health, like in a health environment, is antifibrotic, which means it will regulate the fibroblast. And his target of action is SMAD7. And the effect is the regulation of fibroblast. However, his status in IPF is found to be upregulated. So it means that his antifibrotic effect is even higher. So it's considered an antifibrotic agent. But if we see an example like MiRNA 184, which has been studied in a cell line, it is found to be downregulated. And his function in health usually is to be antifibrotic. However, as it is as it is downregulated, it can fulfill his function. So there will be an increase of TGF-beta signaling pathway, which will lead to an epithelial mesenchymal transition, collagen deposition, fibroblast transdifferentiation, because it acts directly on SMAD2 and the AKT signaling pathway, which is a non-SMAD dependent, which is a SMAD dependent pathway. And in the last position, we can find MiRNA 200, which is one of the most studied MiRNAs nowadays. And it was studied in a bleomycin murine model. And his function in health is found to be antifibrotic MiRNA. However, his status in IPF is found to be downregulated. So we will find an increase in epithelial mesenchymal transition. And the interesting thing in these studies is that MiRNAs can act both in receptors and proteins, cytoplasmic proteins, and also in signaling proteins. I mean, not also in receptors, but in cytoplasm proteins. And here we have another example, which is MiRNA 130B3P, which was found in human lung fibrotic samples. This one was studied in human. And his function in health is found to be antifibrotic. However, his status in IPF is found to be downregulated, which means there will be a poor regulation of fibroblast activation and epithelial mesenchymal transition. And his target of action is IGF-1, which is an activator of the TGF-beta signaling pathway. And we have many, many examples of these MiRNAs. However, these are the most relevant one because they act both in receptors, activators, or cytoplasmic proteins in a SMAD-dependent way. And also we have MiRNAs involved in a non-SMAD-dependent way, which was the other signaling option of this in IPF. So here we have, for example, MiRNA 448, which was studied in a bleomycin murine model, and his function in health is found to be antifibrotic. However, his status in IPF is found to be upregulated. I mean, it will cause an JNK activation, and it will increase cellular, I'm sorry, it will cause increased cellular apoptosis, decrease of cell viability, and reduce collagen synthesis by fibroblasts. So it means it's antifibrotic MiRNA. And also we have MiRNA 29C, which was studied in mice and human lung tissue samples. This MiRNA is found to be it has a antifibrotic function, and it acts directly on FOXO3A. However, his status in IPF is found to be downregulated. So this MiRNA, even though it has an antifibrotic function, it will create a profibrotic environment because it's downregulated, and it will as a result, there will be a decreased cellular apoptosis. So fibroblast will have a longer life to continue with the process of fibrosis. And well, as a last example, we have MiRNA 124-3P, which was studied in a bleomycin murine model, and it acts directly on axin 1, which is a cytoplasmic protein located in a non-SMAD signaling pathway, and it's found to be profibrotic because it will induce TGF-beta1 signaling pathway, and it will cause fibroblast differentiation and increase WNT pathway, which is a non-SMAD pathway, and it is found to be upregulated in IPF. This will be better understood in this template, because here we have the crosstalk signaling pathways, because all of these signaling pathways coexist together and at the same time. And for example, here we have MiRNA 877-3P, which is found to be upregulated. And this, the function of this MiRNA is to regulate the SMAD6 and 7 complex, which causes which causes a regulation of SMAD2 and 3 and 4 complex. If you remember, this SMAD2 and 3 complex is the one who is in charge of secreting collagen, and also causes fibroblast differentiation and proliferation, and the expression of profibrotic genes. So if this MiRNA is found to be upregulated, it means that it will have an anti-fibrotic function. So it will stop the fibrotic process. Also, we have here the MiRNA 1343 MiRNA, which is found to be upregulated, and it has an antifibrotic role because it inhibits because it acts in the TGF-beta1 receptor, and it stops it inhibits indirectly the SMAD2 and 3 complex, because if this receptor is upregulated, the signaling pathway can't follow the processes in the cytoplasm and in the nucleus. And also we have MiRNA 184, which is found to be downregulated. And for example, this MiRNA also, it's an antifibrotic MiRNA. His status in IPF is found to be downregulated, and it will cause an increase of TGF-beta signaling pathway, because this MiRNA will not be able to regulate SMAD2. And also this MiRNA acts in AKT, which is a non-SMAD mediated signaling pathway. So this will lead eventually to the epithelial mesenchymal transition with the transdifferentiation of fibroblast, which will generate the synthesis of collagen. So there are a lot of MiRNAs, as I said. Some of them can act in a non-SMAD mediated pathway, and some of them can act in a SMAD mediated pathway. And some of them have an antifibrotic role, and some of them have a profibrotic role. But their upregulation or downregulation will determine if they collaborate with the fibrotic process or they stop this fibrotic process. As conclusions, we have that MiRNAs up and down regulation can favor the epithelial mesenchymal transition, which will lead to the synthesis of collagen, fibronectin, and also fibroblast proliferation and transdifferentiation to myofibroblasts, which are the key cells involved in this fibrotic process. And also MiRNAs can regulate receptors and cytoplasmic proteins of different signaling pathways, which coexist all of them at the same time. And if we could know more about these MiRNAs and how they act and his state in IPF, if they are found to be up or down regulated, we could use them as future therapeutic targets, because we could silentiate a profibrotic MiRNA or to activate an antifibrotic MiRNA. And we could offer a better treatment for this patient, because this disease doesn't have a cure nowadays. So they will offer them a new therapeutic option and a better quality of life. So further studies are suggested on this field, so we can in the future offer a new therapeutic option for these patients. Thank you for your attention.